All right, what's going on? We're going to get back at it. So we're going to talk about membranes. This is actually chapter 7 in the new textbook, chapter 8 in the old textbook. I haven't got around changing the PowerPoint slides yet. So we'll talk about like kind of a little history behind it. So there, people have known that we, science has known there are membranes, obviously, in cells for a number of years, probably going all the way back to when they began using microscopes. Um, and they knew some basic things about it. They knew that it controlled things going in and out of the cell. They use this term selectively permeable, which means that um, it allows some things in while keeping others out. And they knew some of the pieces that made it up. And they knew that the membrane was not a rigid structure, that it was fluid and is kind of movable. So back in the early 1900s, this is when they discovered phospholipids. Okay, and they were able to see how if they broke cells up and took the components of them and dropped them in the water, what would happen is you would get this layer like this on top of the water. And what's happening is they were able to say that part of it was hydrophilic, part of it was hydrophobic. So they knew this. They had this idea that that part of it didn't like water based on how it separated and stayed in the water. All right. They also found benzene, which benzene is a nonpolar substance that when you mix these, they would actually flip the opposite way. The nonpolar tails would actually end up in the benzene, which is kind of interesting. So the first thought was, and once again, they knew some of the major components. They thought that the membrane kind of had this double bilayer, or the lipid bilayer, and then it was just layered with proteins on top, almost like a sandwich. So you think of like the protein would be like the bread of the sandwich, top and bottom, and then the meat or the ingredients would be the phospholipids, which we know now is not true. We know that these proteins are actually embedded within the membrane. So this was once again early 1930s. So as technology got a little bit better, they were able to look at microscopes differently. A lot of this was based on the introduction of electron microscopes back in the 1940s and 50s. So what they realized here, let's fix this a little bit. What they realized here was that the membranes, cancel. So we're fixing that right now. So what they realized was that the membranes actually, by looking at, excuse me, by looking at the membranes under the electron microscope, that these proteins are actually embedded within the membrane, not laid across the top. All right, which then gives you areas. For example, the proteins tend to be very hydrophilic, the outside of the protein, where the center of the protein is hydrophobic. So basically, we talked about the proteins being made of coils and sheets. So this center lighter section here, it's kind of hard to differentiate. That part of the protein, the reason it's embedded in there is because it's got R groups, or those side chains, that are hydrophobic or water-fearing, and the hydrophilic ends kind of stick out into the intercellular space. Another way they were able to tell this is they were able to freeze a cell and use a special microscopic knife and actually peel the membrane in two. And what they found is that when they froze it and peeled it, they were able to see that there are these proteins that literally were stuck through the membrane. So it's been a as like I said, as technology is getting better, we've been able to do a better job of actually looking at how these membranes function. All right, that's it for this really quick one on the history behind it. You won't see much of this on the AP exam. I just think it's kind of it's a good look at how technology has changed our understanding of things. The term they give this now is, excuse me, is the idea of this fluid mosaic. Fluid meaning movement, and from your art studies, I'm sure, a mosaic is basically an image that you take a lot of small pieces and you glue or paste those small pieces together to then give an image. You see it in a lot of I want to say like Italian, Mediterranean type art. They'll do something called a fresco, and a fresco is basically little bits of glass or porcelain or tile, and they'll take those little colored pieces and attach them, affix them to a wall to then make an image. So that's what a mosaic would be. All right, we'll move on to the next one.